Hello, my name is Dr. Eric DeJesus, founder and CEO of Annate Bitherapeutics. The mission of Annate is to harness the power of the human innate immune system for the treatment of multiple myeloma, and hopefully many other cancers. First, what is multiple myeloma? Well, it's cancer of the plasma cells, meaning that it's a malignancy of the bone marrow that will soon eventually affect immune cell function. It represents about 1% of cancers and 10% of all blood cancers. There are over 35,000 new cases diagnosed each year, and with an aging population and improved diagnostics, has an increase in incidence rate of 8%. The current standard of care therapy begins with stem cell transplant eligibility. From there, patients are treated through a combination of proteasome inhibitors, immunomodulatory drugs, and monoclonal antibodies. As the disease progresses, newer therapies such as CAR-T, vaccines, and antibody drug conjugates need to be introduced because unfortunately, the reality with these approaches is that over 85% of these patients will experience two outcomes: relapse and resistance to those new therapies. At that stage, overall survival drops to just 11 to 23 months. So despite these significant advances, multiple myeloma is still an incurable disease. So the need is clear. We need a next-generation platform that is safe, effective, and utilizes a new mechanism of action. With our approach, we do exactly that by harnessing a naturally occurring component of the human innate immune system to fight cancer. In the bloodstream, high-density lipoproteins, HDLs, often carry a unique protein called apolipoprotein L1, or ApoL1. Historically, our co-founders' research showed that ApoL1 can specifically bind to and kill parasites, revealing its role as a cytotoxic agent in the immune system. This sparked the question, could ApoL1 be used to selectively target and kill cancer? To test this, we engineered a bispecific antibody designed to bind both ApoL1 and cancer cells, specifically multiple myeloma in the blood. With this strategy, we aim to reposition ApoL1 as a precision therapeutic, transforming its innate cytotoxicity into a targeted weapon against cancer. Critically, in our preclinical data, this approach is working. Treatment led to a 40% increase in total cell death in just under 3 days, a strong signal of therapeutic potential. Equally important, our therapy is highly specific. As we've observed, there is no detectable off-target toxicity. Additionally, there are several advantages to our platform, including no need for immune cell recruitment, no addition of external toxins such as antibody drug conjugates, and because of its novel mechanism of action, our bispecific antibody has the potential to be synergistic with the current standard of care therapies. Finally, since it targets native features of both the cancer cell and the HDL, there is no need for personalization and can be considered off the shelf. Naturally asked, could this strategy be used beyond multiple myeloma? By redesigning our therapeutic, we extended our approach to the solid tumor setting, specifically pancreatic cancer. What we were able to observe is that our preliminary treatment data revealed a 40% inhibition of tumor growth. So when combined with our multiple myeloma results, this underscores the potential of our platform as a next-generation therapeutic, capable of targeting both liquid and solid tumors. Even more compelling, these biomarkers targeted by our two leads are shared across more than 10 different malignancies, broadening both the therapeutic impact and market opportunity. But for now, let's bring the focus back to multiple myeloma. Today, approximately 120,000 patients in the United States are undergoing treatment for multiple myeloma, with over 50% experiencing relapse. Current treatment costs translate to an addressable market of $18 billion, with an annual growth rate of 8%. By 2035, that market is projected to be $32 billion. Based on our preclinical data and competitive positioning, we believe that a realistic market share of 10% is achievable, representing a potential $900 million in annual revenue. Now, given this significant potential, what's the precedent for companies at clinical phase 1, well before FDA approval? An analysis of comparable biotech deals shows early-stage acquisition and partnerships valued between $680 million and $2.5 billion, even at phase 1. These deals underscore the opportunity of substantial value creation and investor return well before commercialization. Of course, executing on this opportunity requires the right team with the vision, expertise, and commitment to deliver, and that's exactly who we are. Our story began with the research of Dr. Stephen Haydig, who dedicated nearly 50 years to studying African trypanosomes, including their interactions with APOL1. My involvement began when I posed the question of whether we could pivot APOL1 from targeting parasites to targeting cancer, leading the initial studies on APOL1 toxicity in mammalian systems. However, it wasn't until Steve's wife, Anne, was diagnosed with multiple myeloma that our research took a deeply personal turn. After her passing, our resolve intensified. Dr. Michael Cipriano joined the team, bringing critical insights that led to the transformative idea of targeting APOL1 with a bispecific antibody. Together, we formed the core team committed to advancing this innovative therapeutic platform. Since forming, we have been able to bring on several critical members, including scientific and clinical advisors from the Winship Cancer Institute of Emory, as well as 2 seasoned strategic business advisors. Together, our team possesses strong experience bringing technology out of the university environment with VC money, strategic planning, and executing IND-enabling studies for gaining FDA approval. To date, we have made sustained progress over the last 4 years and have had several key wins. We have continued to secure funding through 3 pilot grants to develop our proof of concept against 2 indications, filed 2 patents on the technology, and have continually advanced our technology forward with an increasing scope in experimental systems. Also, we are constantly applying to foundational grants and SBR opportunities to continue pursuing the non-dilutive side of our funding. What we're offering today is an opportunity to pair up with us at Annate. This will be through a $3 million seed investment, which will help achieve several tangible milestones. Those include finishing optimization of our lead therapeutic and refining the second, performing key experiments to have closer engagement with the FDA, and develop key partnerships to further the development of the IP towards IND submission. In review, here at Annate, we have generated a next-generation therapeutic platform for the treatment of cancers with a novel mechanism of action and a proven delivery system, the bispecific antibody. We have shown that it is applicable to both liquid and solid tumors, and the 2 leads alone can potentially treat over 10 different malignancies. Lastly, we do hold the option for the exclusive licensing rights to the IP, which is owned by the University of Georgia Research Foundation. Our safe note opens in Q4 of this year with a target of $3 million. This will help achieve milestones previously mentioned, but quickly include optimization of our lead therapeutic, validation of the approach in vivo animal studies, IND preparations, and execution of the full licensing agreement. So thank you for the opportunity to share our story today. We're deeply committed to advancing this technology, not only for its transformative potential in cancer research and treatment, but also for its strong commercial opportunity it represents. We look forward to beginning a conversation today on how we can partner to make this vision a reality. Thank you.